Good morning, everyone, and welcome to today's webinar, exploring the European Pharmacopoeia from a microbiological perspective. My name is Aletia Cruz. I'm a microbiology product manager here at CondaLab, and today we're going to talk about the latest edition of this very important document for drug control, especially, of course. I am waiting a few more minutes so people can join us. But let's begin because we have a lot of information. Okay, so I'm gonna go slowly right now, and then we're gonna get into the more important key points. So what are we going to review today? We're going to talk, of course, about pharmacopoeias and their importance in microbiological quality control in medicines, the different types of analysis that we can find in medicine production, in drug production, from the control of an essential raw material such as water for manufacturing and its quality control needs according to its classification. Also, the control of both sterile production process and the final product, depending on the type of pharmaceutical product. Environmental monitoring for good control of cleanliness and good practices. And finally, a part on chapters of the Pharmacopoeia that are interesting, such as the differences that may exist between the USP and the European Pharmacopoeia, although these are harmonized. Um, we are going to talk a little bit about the need of neutralizers in certain analysis and the use of alternative methods. So um, I have a little bit of allergies, so bear with me if you hear my voice kind of weird. It's because of that. I see a couple more people have joined us, so I think we are good to begin. So I'm going to begin with this first part, that is the introduction to pharmacopoeias, what these documents are and their importance. So as you know already, I guess, it is a document that establishes the minimum quality standards that medicines for human and veterinary use, as well as their components, both active ingredients and excipients, must meet in the countries of the Convention of the Preparation of a European Pharmacopoeia of the Council of Europe. This is a definition according to the Spanish Agency for Medicines and Medical Devices. So, according to the World Health Organization, the central objective of pharmacopoeias is to protect public health through the creation and provision of public standards to assist in the quality assurance of medicines. As we see in the image, the history of pharmacopoeias begins well before the 19th century, and there are more than 40 published pharmacopoeias, hence the need for harmonization, especially in the current context of globalization. The three pharmacopoeias that are an international reference are members of the Pharmacopoeia Discussion Group, are the European, the American, and the Japanese pharmacopoeias. However, very recently, this group has added a new member, the Indian Pharmacopoeia Commission, something that had not happened in more than 3 decades. And this highlights, of course, our globalization, our context, current context, and the recognition of the Indian pharmaceutical industry as one of the main manufacturers and therefore an international reference for medicines from now on. As I mentioned before, pharmacopoeias serve as a reference guide for safety standards and controls for pharmaceutical substances and products. And more strictly, from a microbiological point of view, its role is to ensure the safety and effectiveness of medicines by establishing the standards for the control of microbial contamination, sterility testing, and microbiological limits for these products. Since a microbial contamination could mean a very serious risk to the patient's health, even with long-term effects or even in some very serious cases, death. Microbiological control will be based on ensuring an absence or presence, that is, when sterility is required in the product, or if it is not required, it will be necessary to control the microbial load and verify that the established microbiological limits are met. Excuse me for 1 second. Okay, so for the microbiological analysis of these products, these pharmaceutical products, we can establish 5 major pillars that include the analysis of an essential raw material, such as the water for manufacturing, the control of the aseptic process, also the control of final product, where there is a great division depending on the product sterility requirements, and finally the environmental control in the different stages of the process, not only production but even packaging, for example. So we're going to address in a very general view each of these pillars. We're going to begin with the control of water for manufacturing in pharma. The categorization of water for manufacturing will be divided based on sterility requirements and other uses. This will apply also on the final product control that we will see later. For sterile water, we have two categories. The first one is water for injections. That is water that meets the same requirements as water for consumption and is produced through distillation or purification processes such as reverse osmosis. For microbiological monitoring, measures are taken to ensure that the microbial count is adequately controlled and monitored, of course, and under normal conditions, the microbial count has to be of 10 CFU per 100 milliliters. When performed by a membrane filtration, this membrane with a pore size less than 0.45 micro. For aseptic processes, of course, that stricter levels could apply. On the other hand, we have sterile water for injections, which is the previous water only packaged and sterilized by heat under conditions that ensure compliance with the test for endotoxins. For microbiological monitoring, also you must comply with the sterility test. We're going to move on with water for manufacturing for other non-sterile uses. First, we have purified water prepared by any suitable method that meets the requirements of water for consumption, such as reverse osmosis, ion exchange, distillation, etc. For biological monitoring during production and storage, measures are taken to ensure that the microbial count again is controlled and monitored. And under normal conditions, the microbial count has to be of 100 CFU per milliliter when determined by membrane filtration. Then we have the same type of water, but in containers. That is, it is packaged and stored in a way that meets the conditions to ensure its microbiological quality. Microbial monitoring in this case is the count of total mesophilic aerobes with an acceptance limit of 100 CFU per milliliter. And then finally, we have the water for the manufacture of extracts, specifically herbal medicinal extracts, are indicated, and it meets the requirements for purified water or water for suitable for consumption. Microbiological monitoring under normal conditions is the microbial count of 100 CFU per milliliter, again determined by filtration. In the past editions of this document, the European Pharmacopoeia, another classification was included for water for manufacturing, that is the highly purified water, which was indicated for the preparation of medicinal products in which a high biological quality was required. However, it has been since eliminated because it was the same microbial monitoring as the one indicated for water for injections. So let's continue with the microbiological control depending on the type of water that we've seen. Um, as I said, on one hand, we have a stricter control for certain types. For injectable and purified, the control purified water, the control will be carried out using R2A agar, incubating between 30 and 35 degrees Celsius for at least 5 days. R2A agar is a medium that stimulates the growth of stressed or injured bacteria that are tolerant to chlorine thanks to its low nutrient content and low incubation temperature for a long time. So before carrying out the water analysis, a growth promotion test must be carried out on the medium, which involves the preparation of reference strains. In the case of this R2A agar, we have Pseudomonas aeruginosa, Bacillus subtilis, very common strains in farm analysis. And as you can see here on the right in our technical sheet, we include, in addition to these strains, others to ensure a high quality of the culture media. We're going to continue with the other type of analysis for purified water in containers and water for extracts. We have the so familiar TSA agar with the same incubation conditions as the previous culture media, 30 and 35 degrees Celsius for at least 5 days. The TSA is a general purpose media that is very rich in nutrients and for the cultivation of both fastidious and non-fastidious organisms, which has a multitude of uses, both for isolation and maintenance of strains. These reference medium, both that we know from ISO standards and as well as Pharmacopoeia that we see here, is going to go through a growth promotion test with all these strains. And again, we can find very common pathogens or contamination microorganisms such as Staphylococcus, Candida, Aspergillus, that are very common pathogens for analysis in the pharma industry. So we're going to leave now behind this topic of raw materials, the water for manufacturing, and we're going to move on with the control of the sterile production process. In this case, I am talking about the aseptic process simulation, APS, also known for a long time as media field testing. Both names are very descriptive of the process. The APS is a tool for evaluating the capability of aseptic process activities using a microbiological growth promotion culture media instead of the pharmaceutical product. So that means that the risk of contamination of an aseptic process is going to be evaluated by carrying out the manufacturing process, but using a culture media instead of the pharmaceutical product. It is necessary in part because the sterility test used to release batches of sterile products has inherent limitations in detecting contaminated units in batches with low levels of microbial contamination due to the limited number of samples that can be removed for destructive testing. All of this that I'm telling you has been evaluated statistically. Um, Hold on a second, please. Okay. So, as I've said, the APS simulates the aseptic process of the product and the sterilization of all components, until the final sealing of the container. The culture media is going to make contact with all contact surfaces of the product processing equipment, containers, critical environment, and the same manipulations to which the product is usually subjected to. It is incubated to later inspect it and verify the presence or absence, obviously, of microbial growth. Ultimately, this information is going to be used to evaluate possible contaminated product units during the aseptic processing operations. So, in short, the APS challenges the capacity of the aseptic process, but not only the production process, but also to produce sterile products in a reproducible manner, evaluate the process contamination risks, detect any vulnerability to microbial contamination, monitor compliance with good manufacturing practices and regulatory expectations, and to demonstrate that these practices and processes are appropriated. Also, the staff and their competencies are evaluated. As for the culture media used for the APS, we're going to find TSB broth, which is again we are all very familiar with it. It is a very general purpose culture media that allows the growth of gram-positive, negative bacteria, mold, yeast, fastidious microorganisms, and some anaerobes, even at very low levels. So within the TSB formulation, we find certain compounds, such as, for example, the sodium chloride, that is going to supply essential electrolytes for transport and osmotic balance, and some other agents. This culture media is going to be um, incubated for 14 days, and at the end, there should be no growth of any kind. The APS requires three controls: process control, negative, and positive controls. So, how frequently should an APS be performed? That really is going to depend on the risk assessment of the aseptic process, and also situations such as validation of a new manufacturing facility or site on a semi-annual basis for already validated lines and processes, or when a major change occurs in an aseptic facility or process. It is evident that the more APS are carried out and with periodicity, the greater control there will be over the aseptic process. So if you want more information about the APS, we have a webinar only on this topic. We have it on our website. You can find the link to the recording. So let's move on to the to part 4 on final product control. Um, Here in Europe, we're going to find different drug regulatory bodies. On one hand, we have the European Medicines Agency, EMA, the body responsible for the evaluation, supervision, and monitoring of the safety of medicines in the EU. The European Commission also adopts regulations, legislation, and guidelines related to pharmaceutical regulation, and then the competent national authorities of each country for the evaluation of medicines, inspection of facilities, and also for the surveillance of adverse reactions to products. Here in Spain, for example, we have the Spanish Agency for Medicines and Medical Devices that I mentioned earlier with the Pharmacopoeia definition. We have also key guidelines and directives, good manufacturing practices within the EU for ensuring the consistency of manufactured medicines and the control of quality standards. good clinical practices, guides that govern the clinical trial process to ensure right safety and well-being of the participants, pharmacovigilance within the EU, which is a very robust system to monitor the safety of authorized medicines, and finally, our star of the webinar, the European Pharmacopoeia, which provides official standards for the quality of medicines and their components that must be legally complied with. So, medicines are going to be divided into two types depending on their microbial quality requirements. We have on one hand the sterile ones, and on the other hand the non-sterile ones, very similar to the categorization of water for manufacturing. For the former, sterility is achieved by terminal sterilization and or through an aseptic manufacturing process where the APS enters. For the non-sterile, it is just a control environment, of course. In sterile, the environmental control requirements are very strict. There are classified clean rooms, and in non-sterile, there are unclassified clean rooms, so the environmental control is not very strict. The microbiological analysis is carried out by testing the sterility of the product, and there must be an absence of microorganisms. In non-sterile cases, the total microorganism count and detection of some specific pathogens are performed. Sterile products are, for example, parenteral injections, products for sensitive issues, and the rest of them would be non-sterile products. So, let's look at examples for each type of product. We have here for sterile ones, such as injections, of course, the most common one. Also eye drops, irrigation fluids, cytotoxic medications, and parenteral nutrition. Usually when we think about sterile pharmaceuticals, we think about injections, but there are also some more common ones, such as the eye drops, that usually we don't think of them as having such a strict microbiological or quality control, but they do. For non-sterile ones, we're going to find solids, tablets, capsules, powders, liquids, syrups, suspensions, including aerosols, very important, semi-solid creams, gels, etc. And then another type of drug delivery system, we have transdermal patches, osmotic pumps, etc. So now let's address in a general way the analysis to be carried out by the final product type. For the sterile ones, we have the sterility test. For this, we just simply go through, we put the sample through a membrane filtration or even a direct inoculation, depending on the product. If it's liquid, semi-solid, solid, aqueous, oily, so it is important to take this into consideration. For the analysis of aerobic and anaerobic bacteria, we use TFM, thioglycolate fluid medium, and for fungi and aerobic bacteria, TSV broth. Incubation between 30 to 35 degrees Celsius for a period of no more than 14 days. No turbidity should be observed, that is, no growth, to comply with the sterility test. And then in the case of non-sterile ones, it is different, since we have the total count of aerobic mesophiles, mold, yeast, and then the detection of a specific pathogens depending on the type of product, such as E. coli, Salmonella, Candida, Staphylococcus aureus, and Pseudomonas aeruginosa. Each of these pathogens has a method and a selective media, but sample preparation is very important, and depending on the analysis, TSB broth or buffered Peptone water can be used. And if the product has preservatives in its formulation, neutralizers can be added in this step. So if anyone is interested in learning more about each method, we have webinars detailing each type of analysis. And as I mentioned before, you can find the links to the recordings on our website. For this analysis, at Conda Lab, we have developed the line of LC plates, which include two concepts: long shelf life, since Petri dishes usually have a maximum life of 3 months from their manufacture, and the lateral code, which is the lateral labeling for user comfort and to facilitate the reading of the plates. We have all of these culture media available. So if you are interested in any of them, please let us know. Um, Again, for we have all the necessary products and in different formats for your comfort, for the control of the final product of these pharmaceutical products, both sterile and non-sterile. The media that we have mentioned: TSV broth, thioglycolate fluid medium, and non-sterile selective media such as the cetrimide for Pseudomonas, McConkey agar for E. coli, XLD agar for Salmonella, etc. We have all of these products in dehydrated and prepared format, ready to use plates, bottles, tubes. We have had an experience with some clients in the pharma industry using TS bags for their analysis, which we find more in the food industry. But hey, if it works for them, so it is available. Um, we are already at the last couple of points of the webinar, and the first of them is the environmental monitoring. So sometimes I think that we tend to focus more on the contamination of a final product than on prevention throughout the entire process. For this, the environmental monitoring is necessary since there are very strict regulatory requirements at drug production and handling sites. Good environmental monitoring is going to impact the assurance of quality, effectiveness, and consistency of products, also complying with the standards of regulatory agencies, preventing and being proactive in possible contaminations by finding probable sources is crucial in the validation of aseptic processes to warranty the absence of microorganisms. It helps to better manage risks and prevent the costs associated with an incidence of a contamination, and also to guarantee that the product is safe until it reaches the patient, preserving the medicine, the pharmaceutical product, and finally, thanks to all of these above, protecting the health of consumers and avoiding risks to the general public health. We can categorize environmental microbiological analysis into two. First, air control by sedimentation and surface control. For the first, Petri dishes with general media such as TSA agar or SDA agar are used and can be irradiated with double or triple bags depending on the sterility requirements. On the other hand, for surface control, you can also use 90 millimeter Petri dishes and with a swab collect the sample from the surface and rub it on the plate. Also, there are Rodak plates for direct sampling of the surface. We can find more selective and also general media. Neutralizers are usually included in the formulation to inactivate the surface cleaning agents. These can also be irradiated with double or triple bags. At Conda, we have an exclusive line that was developed for environmental monitoring in pharma. These are Rodak plates that have a special closure to avoid cross-contamination due to accidental opening. The transportation from the sampling location to the laboratory is safer, and also these have a lateral, here we can see it, a lateral labeling to make it easier to read the results. The lock and block mechanism is very simple. Just by turning the lid, we have two closing positions. Close that you might be able to read here on the plate. Close for an hermetic lock, and air here on the other side for a minimum opening for incubation, and with the side labeling, as I said, to facilitate the reading of the plate. So we are at the almost at the end of the webinar with our last key point about additional chapters that we can find in the European Pharmacopoeia. So I mentioned during the introduction that there are 3 harmonized pharmacopoeias and references at an international level. 2 of these are the American Pharmacopoeia, the USP, and the European Pharmacopoeia, the EP. However, there are very small differences between these two. The USP includes, in addition to medicines, standards for um, sorry, standards for supplements and food ingredients. The European one is exclusively about medicines and their products. Um, the USP includes methods and analysis unique to the US regulatory environment, such as the Burkholderia cepacia analysis in Chapter 60, and the APS is indirectly included in the pharmacopoeia, if you can say so. That is, we can find it in the pharmaceutical good manufacturing practices of the FDA, which is the drug regulatory entity in the USA. And on the other hand, in the EP, it includes tests specific that align with European regulatory requirements and their agencies. Moving on, we regarding antimicrobial preservation in pharmaceutical products. If a pharmaceutical preparation does not possess inherent antimicrobial activity, antimicrobial preservatives may be added, particularly in aqueous preparations, to prevent or limit microbial contamination that may occur during normal conditions of storage and use, especially in multi-dose containers that would present or that could present a risk to the patient. It is important to mention that the addition of a preservative does not replace the GMP, the good manufacturing practices, and that in consider that the effectiveness of the preservative can be increased or decreased by some active ingredient in the product composition or even by the packaging used. The activity of the preservative and its final packaging is going to be investigated during the useful life of the product to ensure that said antimicrobial activity is not going to be affected by the storage. Some microorganisms used to test the preservative effectiveness are common contaminants in medications such as Pseudomonas, Staphylococcus, Candida, and Aspergillus. And finally, we are going to talk a little bit about the alternative methods for drug analysis. So, classical or traditional microbiology, despite being the reference in many international standardized methods, such as pharmacopoeia, as we've seen, or ICE standards, lack speed when it comes to obtaining results. For many, many years, the use of alternative methods has been sought as a response since, among other things, due to the slowness of these results, they prevent us from reacting quickly and implementing corrective measures in the short term. Therefore, there is a need for rapid methods in the analysis of medications, not only for the final product, but for the process, the environmental monitoring, among others. However, the pharmacopoeia still does not recommend one method or another, but instead indicates just the available methods or techniques which could be implemented as a supplement or even alternative to the reference method, which are the culture methods. In this chapter on alternative methods in the pharmacopoeia, we can find the main types of microbiological tests indicated, which are, of course, qualitative with absence or presence, quantitative for accounting, and identification analysis. The growth-based methods mentioned include electrochemical methods, gas measurement, bioluminescence, turbidimetry, and growth detection in selective media. For direct measurement, it indicates solid phase cytometry, flow cytometry, depth, direct fluorescence filtration, and autofluorescence. And for the analysis of cellular components, phenotypic and genotypic techniques. Regarding the validation of these alternative methods, the Pharmacopoeia indicates the steps to follow to validate it, which include the description of the technique, the risk-benefit analysis, a primary validation, and consequently a validation for the intended use. The validation processes will depend on the type of microbiological analysis. And here I leave the other webinars that we have done on the pharma testing series, where we go into, as I mentioned, more depth about the analysis of both sterile and non-sterile products and the APS. We have a brochure with the workflows according to Pharmacopoeia, which I must mention we are working on a new edition and that we hope to publish very soon. I'm going to end the presentation here, and I'm going to move on to the questions that have been sent to us. We have just a couple of minutes, so I'm going to try to answer the most important email to the people who have sent these questions. So let's see. Okay, we have a lot of, as I mentioned, you can go to our other webinars. I see people interested to go more into the analysis of sterile, non-sterile products. So if you can go, please go watch those webinars. You have all that the analysis described there. Okay, let's see what we have. Why are many more why are many more microorganisms included in the analysis of non-sterile than for sterile? Okay, this is very easy for me to answer. That is because for sterile products, non-sterile products, sorry, a specific pathogen or indicator is going to be sought. And for sterility, you are looking for absence presence. So that is, in sterility, you cannot have any type of growth. And for this reason, general media are used because they support the very wide range of microorganisms. So that is the difference. In non-sterile analysis, is the use of neutralizers mentioned? Which ones can be used? Okay, I mentioned during the final product control for non-sterile analysis that in the preparation of the sample, you can add neutralizers. Also, you can do it directly in this step, or also to the culture media. However, I think it would be best on the preparation of the sample. You can use agents, neutralizing agents that are very common, such as lecithin, polysorbate, twin, there is the commercial name, thiosulfates, and there is a table in the Pharmacopoeia that you can refer to. And depending on the type of preservative, you use a neutralizer or another. So, but in some cases, even a good dilution process is sufficient. So, how often should a media fill be performed? So, media fill or the APS, you don't have a standard. As I said, it depends on the situation. If you're doing it on an already validated production line or site, if that production line of site is new, or if major changes have occurred, I think it's every 6 months. Probably that would be the minimum, but there is not a specific number because it depends on the situation. Um, can an alternative method become a reference method? Yes, of course. Um, the reference method is only the reference method because it is the one indicated in the pharmacopoeia or in the ICE standard, for example. It does not mean that a reference method is always a culture media method. That is just the case at the moment. That both in ISO and in Pharmacopoeia, both reference methods are by culture media. However, in the future, if an alternative method proves to be better than the ones available, then that and it is included within the Pharmacopoeia, so that would become then the reference method. So we have some other questions. I think I have answered most of them. You're going to I'm going to answer the rest by email during these days. I hope you have enjoyed the presentation and that it was helpful. I'm going to remind that you were going to receive the recording and the certificate of the attendance tomorrow to the email addresses which you signed up with. And as I always, I'm going to please ask you to follow us on LinkedIn and subscribe to the newsletter. And these are the ways in which we inform you about any news at Condolab. So for now, I say goodbye and until next time.